Dear viewers, dear listeners, dear ladies and gentlemen, we are very welcome Dr. Björn Brembs to our series of the video interviews that we record here. Dear Dr. Björn, could you please share more about the research that you are doing in your research laboratory? What are your goals and what is the mission of your lab?
Yes, hello. Thanks for having me. I'm a neurobiologist, so I'm interested in how brains organize behavior. And with brains, I mean nervous systems or brains that belong to nervous systems from all kinds of animals. So I'm interested in and humans, of course, as well. And so we use all kinds of animals, mostly for the time being. We use the fruit fly Drosophila, but we have also used leeches or marine sea slug, Aplysia california. We're interested in how nervous systems organize behavior. That means, how do they decide what to do next? And nervous systems usually generate activity all the time. So nervous systems are always active. And so one of the questions that we have is, how do nervous systems pattern that activity, even if there's nothing in the environment that tells them what to do? So how do they spontaneously generate activity? And in terms of behavior, how do they generate actions? And because this is a very rare case that there are no external stimuli, most of our experiments are actually concerned with how the animals then evaluate the feedback that they get from these actions. And nominally, those are situations in which the animals are placed into a novel environment where they will get feedbacks that we control and that the animals have not encountered before. So we study how the animals learn how to deal with this feedback. And this is analogous to, for instance, how young children learn to articulate language. And so they start to babble and they get auditory feedback. So they hear what it is that they're saying. So as everyone knows, all young children want to say daddy first, right? That's the first word that obviously every child wants to say. Every dad can attest to that. And so they start with da, 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 and then what they're trying to say is daddy, and they hear that what they're saying is not quite the same, and so they have to try out how to move their mouth, how to change the speed and the way the air goes through the larynx and these sorts of things. And nobody can tell them how to do that. So there's no instruction involved. So the children have to try that out by themselves. And we do try to design experiments where also the flies have to try things out. So they have to spontaneously generate actions and then evaluate the feedback that comes from that. And why am I talking about humans and flies in the same process, or about this process? And that is because there are genes involved in these type of learning processes that are common to humans and fruit flies. So for instance, the human FOXP2 gene has an ortholog in the fly, the Drosophila FOXP gene. And this feedback-based learning is disrupted both in patients with a mutation in that gene and in flies where this gene has been mutated. And while the other possible learning mechanisms are unaffected. So the humans show normal intelligence and they can learn all kinds of things just fine. They just have articulation problems. And the flies can learn all kinds of stimuli such as odors or colors or patterns. They can learn those just fine. It's just the feedback-based motor learning, if you will, that is blocked. So that's the main focus of our lab, is that how do the animals generate actions and how do they learn from the consequences of these actions?
Sure. Which kind of methods do you use to record these activities and to distinguish the differences between different patterns or behaviors?
So the machine that we use most of the time is a so-called torque meter, where the flies get attached with a drop of glue to a little hook, and that hook then can a measuring device. And that measuring device measures the attempts of the fly to fly to the left or to fly to the right. So to rotate around its vertical body axis. And this is, of course, quite tiny. These are quite tiny forces. Those are in the pico to nanonewton meter range. So that's 10 to the minus 9 to 10 to the minus 12 Newton meters. And so these are machines you cannot buy. So at the moment, we are developing new such machines. Our old ones have run their course, and now we need to replace them. So let's see how that works. I can show the place where this machine would usually be, which is right next to me here in our lab. So you can see this arm, which is now empty. There the machine would be, and then the environment around the fly would be here. The fly would be inside of this, and then here is a camera below. This is also in the workshop right now. There's a laser that would give the animal feedback about whether that was the correct turning direction or the wrong turning direction. And so all of this is in our mechanics and electronics workshop right now to be upgraded. And I hope that within the next couple of weeks, I can go back to doing these experiments here in my room as well. We do have several other behavioral experiments and setups as well for different aspects of the same behavior, but those are like the most used setups that we use in our lab most of the time.
That's interesting. So far what I knew that physicists normally they build their own devices, but that those people who study neurogenetics need to build their own devices. This is I hear for the first time. This is very valuable information, but those devices, they are not that much sophisticated in terms of the mechanical and electrical engineering, or there is still work involved. How do you manage to lead both sides, electroengineering essentially, and the biology in your lab?
So I could not do this alone. So like I said, we do have electronics and mechanics workshops where dedicated engineers collaborate with us in developing these machines. So for instance, for this measuring device, what you have is you have a tiny little laser on an axis that is a tiny little mirror on an axis that is where a laser shines onto that mirror and then gets reflected onto a detector that's right next to or below the laser. And the fly sits at the bottom of this axis, and if the fly attempts to rotate to the right or the left, then it will move that mirror just a tiny little bit. At the same time, on the same axis, we also have a little coil in a very strong permanent magnet. And we use the signal from the detector of the movement of this mirror to feed back current into that coil to counteract the rotation. So the animal actually is not really ever moving. The current that goes through the coil that puts that keeps the axis from rotating is of course proportional to the force that the fly exerts to rotate it. And so this is how we measure the signal, is by generating a counterforce that rotates the axis with the same force in the other direction. And that requires then a lot of, and those are all technologies that I could devise in theory and in principle, but I could never build these things and develop these things with the expertise that I have. And so without the engineers and the mechanics and electronics workshop, I would not be able to develop these devices.
Yes, it is an interesting take-home message that the studies on humans, they are requiring the brain activity, but the studies on the flies, they require the movements of the animal. So this is something very different from if I would study humans. Speaking about the current projects that you have in your research laboratory, how big is your research laboratory? How do you decide which projects to give?
Well, usually, I mean, initially, when I started this research now, what is it, almost 30 years ago, 28 years ago, I essentially asked the questions that I found most interesting. And so I started to look into these feedback-based learning mechanisms and how they were different from other learning mechanisms, such as the learning that takes place when in situations that are similar to how the Pavlovian dog learns about the bell ringing before the food comes. And so those mechanisms require a completely different set of biochemical cascades. So the genes and molecules involved in this type of learning seem to be completely separate from the molecules and genes involved in this feedback-based learning mechanism. And so initially this was not known. And initially everybody thought, well, there's one way how nervous systems learns and how nervous systems learn and how neurons modify or get modified in order to store information. But now we know that there's many different ways of modifying neurons to store information. So at the time, I was just interested in how is that similar or different? And then increasingly, the questions come up by themselves, by the results that we get. So for instance, one research stream that we're pursuing now is, okay, we found this FOXP gene, and we have found through other experiments, we have found a atypical protein kinase C gene, which is also important. So another question is, how do they order themselves? So what comes first? Is the FOXP gene important during the learning, or is it important during development to set up the whole network that then can learn? What are the substrates that this protein kinase C is phosphorylating? And so these questions then come up quite naturally and organically from, you open one door and then you see all these other doors that are still there that need to be opened. And so now in most of the cases, the immediate next questions come quite naturally from the results that we've gotten previously. So as long as the general question, for instance, is how do these feedback-based learning mechanisms take place within neurons and between neurons, then the next smaller questions then come quite naturally and organically.
Sure, thank you so much for this. What is your outlook for the future? What is the prospect essentially? What would you like to achieve in the future for yourself and your research laboratory?
Well, so in this line of questions, of course, we would like to know in the end which neurons are the ones where this plasticity takes place. And as of now, it looks as if motor neurons in the ventral nerve cord of the fly seem to be very important, which seems to fit a little bit with motor areas in vertebrates. This kind of learning has been seen to modify motor neurons in the spinal cord, which is actually in some instances being used in therapy, is to invoke motor learning, spinal motor learning in patients with stroke or spinal cord injury. And so this matches quite well. And so right now we're focusing on these motor neurons in the ventral nerve cord of the fly. And we would like to find out, how are they changed? So are the synapses changed? Is the excitability changed? What channels in the membrane, what biophysical properties are changing and which remain the same? What is the biochemical cascade that leads to these changes? So that's a very sort of near-term research goal. The longer-term research goal that we've just started exploring now is how do these learning processes interact with other learning processes? So like I said, this Pavlovian learning and this feedback-based learning have very different biochemical pathways. But what we do know is that they interact with each other neuronally and genetically. So for instance, if we switch off certain genes here, if we use mutations in this Pavlovian learning, then this feedback in these animals, in these mutants, the feedback-based learning, what we observe is it's increased. These mutants that are deficient in Pavlovian learning are actually better than wild-type flies in this feedback-based operant learning. And so there's a genetic interaction in some way. And on top of that, there's also a neuronal interaction. So what we have found is that if we block certain neurons in the fly brain, then what happens is that this interaction that happens between those two learning processes, and this interaction is that the learning about external stimuli, like the Pavlovian bell, this learning slows down the feedback-based learning, such that if both can happen at the same time, only the Pavlovian learning happens, and we cannot see this feedback-based learning anymore. And only if we train the flies very, very long, then we can see this behavioral learning, this motor learning to come up as well. And if we block certain neurons in the fly brain, then what happens is that this motor learning comes up too quickly, more quickly than it usually would come in wild-type flies. So the long-term goal would be, how is this biochemical cascade in the motor neurons, if this is the main part, how is this slowed down? So how can learning in one part of the brain, one part of the nervous system, which is actually the brain, there we know where the learning happens, how can that learning, if it is going on, how can that influence learning in a completely different place of the nervous system? And so we're currently in the process of finding out which neurons are involved. And once we have found out which neurons are involved and how, then of course we want to find out how this interaction takes place. And of course, because we still don't know how the motor learning itself operates, of course, then it's going to be taking much longer now to find out how this learning then gets modulated. That in some contexts it's faster, and in other contexts it's slower.
Sure. Of course, this neuroorchestra, it is very complex, and it is very interesting to decipher these mechanisms. To our viewers who can be students or they are scientists, as well as to those who are potentially patients, we can also have some viewers who are in the investments, business angels, as well as VCs. To those three groups of people affected by the diseases and those who are sick, what message would you send to them across?
Well, for the people who want to join our lab, of course, they should just contact me if they're interested and let me know what they're interested in, and and then we will see what we can do. I mean, we have set up our lab to be quite flexible, and at the moment, or most of the time, we usually have or can make some space for people who are interested in this sort of research. So this, for them, I would just say, contact me. For the people who are affected by diseases in this aspect, there's not a whole lot we can do. Now, because we are starting to find out which genes and processes are involved, and of course, the prospects of treatment now start to become more apparent. So, for instance, these kinds of motor learning processes that we study are very often used as models for disorders which lead to a problem with behavioral control. So this could range from something like Tourette's syndrome with tics, where you lose control over your behavior. But this could range all the way to drug addiction, where you also lose control over your behavior, where if you're attempting to stop using drugs, you find it very difficult to do that. And so what we're studying is this interaction between two different learning mechanisms. And one of those learning mechanisms is this motor learning that leads to automation of behaviors. And what we're studying is how other forms of learning can slow this automatization of behaviors down, which means that if we would be able to find out how this works on a genetic, neuronal, and molecular level, we might be able to prevent or help slow down the development of tics or of addiction, such that these patients would not be losing the control over the behavior. And for investors at this point, I don't think I can say a whole lot, simply because it is all very basic research at the moment. And at this point, but of course, much of these very basic genetic underpinnings of these processes have been conserved, right? So the reason the flies have a FOXP gene and we also have FOXP genes is because these FOXP genes evolved at some point in the last common ancestors of flies and humans. And apparently the function of these genes was so important that they have been conserved through about 500 million years of evolution. So that means that whatever mechanism we find, at least for now, looks as if it's highly conserved in evolution, which means that any pathways that come out of this research looks like it's might be conserved in vertebrates and in mammals and humans as well. So once we have found out what that pathway is, there's a very good chance that similar pathways take place in the human brain as well. And so if we find out what it is in flies, as soon as that has been established, then of course one can look at possibilities to invest in research that tries to establish if these same processes are taking place in humans as well.
Thank you so much for this. Would there be about it?
There's a little technical aspect to this that I think might be interesting. So all of this research, of course, generates digital data, right? So our machines are computer controlled. Our experiments run mostly automatic. So we have to be there and make sure that there are no problems. But if the flies cooperate, then usually these experiments run automatically. And what we also do is we automatically publish these raw data together with quality control data and statistical evaluations, because we think that open science is the best way to do science, such that everything that we do becomes immediately open and accessible. So the software that we use to control the experiments and then to evaluate the data and do the quality control and the statistics, they're all accessible, they're all open source. The raw data are being published also openly and accessible to everyone. And then when we write our manuscripts, then we also try to write them in an open manner as possible, such that during the publication process, the writing process, everyone can scrutinize what we're doing and try to reproduce what we're doing and catch mistakes, because we're only humans, everybody makes mistakes. And so by making things open and allowing people to have a look at what we're doing, we're trying to be proactive and trying to encourage people to find mistakes as early as possible, because everybody knows how embarrassing it would be once something is finally published in some sort of semi-final way, and then mistakes are found. This is something that nobody really likes to have. And so we're an open science lab, and we're trying to make everything as open as possible without generating too much extra work for us. That's why we've automated all of these things. We've automated the open source development. We've automated our data deposition, such that we can be an open science lab without actually having to invest too much extra effort.
Good. Thank you so much for this. That was our pleasure to learn.